now we have Parool Gupta joining us from India with the ICMR Memorial Research Institute of Medical Sciences, and they'll be presenting on donovani, Cfd1 and Nbp35 proteins and how they make stable cytosolic complex and contribute to the parasite survival and Amphotericin B resistance.
Yeah. Can everyone see my slides?
Yes, we can see your slides. You can go ahead.
So thank you so much, ma'am, and the entire BSI team for providing me this opportunity. So hello, everyone, and namaste from India. I'm Parool Gupta from Indian Council of Medical Research, going to present my research work. Leishmania donovani, Cfd1 and Nbp35 proteins make stable cytosolic complex and contribute to parasite survival and Amphotericin B resistance. In this project, I'm mainly focused on the Cfd1 and Nbp35 proteins, which are from CIA machinery, and they helps into the parasite survival and Amphotericin B resistance. So coming to the introduction part. 1 minute. Yeah. So coming to the introduction part, the disease leishmaniasis or Kala-Azar affects annually 2 million of people worldwide. In India, 90% cases came from Bihar, that is where our institute is situated. So in this map, we can see the state of Bihar, where around 50% of districts are affected with the visceral leishmaniasis and post-Kala-Azar dermal leishmaniasis. And in this graph, we can see that the year-wise incidence of Kala-Azar, which is declining but not eliminating. So the reason behind it is the PKDL patients, because PKDL patients act as a reservoir for the parasite. And in this figure, we can see the life cycle of parasite, where it completes into the two hosts. First one is sandfly vector, where the promastigode form is found, which is a flagellar stage. And the second one is human host, where the amastigode form is found into the macrophage of the human cells. So due to the emergence of drug resistance, there is a need of new drug target and a new drug discovery to kill the leishmania. That's why we are focusing on the iron sulfur proteins, which are found in all domains of life. And we know that the iron sulfur clusters are the cofactor for the activity of iron sulfur proteins. And the iron sulfur proteins are very essential for the DNA replication, DNA damage repair, electron transport chain, iron homeostasis, etc. So there are 4 different machineries for the biogenesis of iron sulfur cluster, in which I am focusing on the CIA machinery, which is found in the cytosol of all the eukaryotes, which is assisted by the mitochondrial ISC machinery. In this CIA machinery, we can see that the CFD1 and NBP35 proteins interact with each other and form a scaffold for the generation of iron sulfur cluster. Then this cluster is transferred to the apoproteins to convert them into the holoproteins with the help of different intermediate iron sulfur proteins. And this box is showing the some examples of iron sulfur proteins which are getting matured by this machinery. So now for the very first time, we identified CFD1 and NBP35, and
Parool,
in the hello.
Parool, your slides are not advancing. I think you might need to switch your. Yeah, there we go. Thank you.
No, it is fine.
I think you might need to switch your screens.
Yeah, full screen. So this is full screen, sir.
Can you go on slideshow and present?
Slideshow.
So if you look at the bottom right corner of your screen, there's a button right next to the number 68%. That would be the presenting button, I believe.
If you have two screens, you may need to go. And
Can I present in this view?
yes.
Yeah. Thank you so much, sir.
Okay.
So,
Thank you.
For the very first time, we identified, cloned, expressed, and purified the Cfd1 and Nbp35 proteins in Leishmania donovani. And the purification was done with the help of affinity column chromatography, and their cytosolic localization was also confirmed with the help of digitonin fractionation method, where it follows these two proteins follow the same pattern as cytosolic reprodoxin, which is already reported in our lab. So I have used it as a control, and the localization was further confirmed with the help of indirect immunofluorescence assay using confocal microscopy, where we can see the green fluorescence in the cytosol of parasite, which confirms their localization in cytosol. So in the introduction figure, we have found that these proteins are interacting proteins. So interaction between these proteins was confirmed with the help of co-purification pull-down assay, which is a in vitro method, and the immunoprecipitation assay, which is an in vivo method. So here we can see the band for both the proteins in elevate fraction in all the methods, which confirms the interaction of the proteins of Cfd1 and Nbp35, and they were also confirmed with the help of immunoblot analysis. And the interaction was also confirmed with the help of in silico analysis. In this table, we can see the amino acid residues which are helping in the interaction of proteins. So interaction was confirmed. Now we want to check the expression of the Cfd1 and Nbp35 at transcriptional level and translational level. For transcriptional level, we performed semi-qPCR where we can see the intensity of Nbp35 as well as the Cfd1 genes are higher in Amphotericin B resistant strain as compared to the Amphotericin B sensitive strain. And this expression was further confirmed with the help of real-time PCR, where the expression of Cfd1 gene is 15 times higher, and the Nbp35 protein and Nbp35 gene is around 25-fold higher than the sensitive one. And at translational level, the immunoblot analysis was done, and this band intensity was shown here in the form of a graph, where we can see the 2 times higher expression of Cfd1 in resistant strain and 4 times around a higher expression of Nbp35 protein in resistant strain as compared to the sensitive strain. Now, to check the role of Cfd1 and Nbp35 proteins in Leishmania donovani, we generated overexpressor strains of parasite, for which we cloned these genes into the PXE-GFP plus vector. After that, the construct was transfected into the wild-type strain, and the transfectants were confirmed with the help of confocal microscopy. And the tolerance against Amphotericin B was also checked with the drug sensitivity assay, and we found that the overexpressor strains are showing higher IC50 value as compared to the wild-type strain. After that, we want to check the effect of overexpression of Cfd1 and Nbp35 on other iron sulfur proteins. So we targeted some cytosolic and mitochondrial iron sulfur proteins, where we evaluated the aconitase activity, succinate dehydrogenase activity, and sulfite reductase activity, and we found that all the iron sulfur proteins activity are increased in the resistant strain as well as in the overexpressor strain, which confirms that the increase in maturation of iron sulfur proteins in cytosol and mitochondria of Leishmania donovani parasites, which increases the scope for the parasite survival benefits and under the stress condition. So now coming to the summary of my findings and future scope, where Cfd1 and Nbp35 proteins are cloned, expressed, and purified, and these are localized in cytoplasm, and their interaction was proved with the help of different methods. And the expression of Cfd1 and Nbp35 genes and proteins showed higher in the Amphotericin B resistant isolates as compared to the sensitive ones, and IC50 value of Amphotericin B was also found higher for the overexpressor strain as compared to the wild type strain. So these results cumulatively implicate the contributory role of Cfd1 and Nbp35 proteins in survival and Amphotericin B resistant of L. donovani. Further, the inhibitors will design against these two proteins and will be screened, and the role of these proteins will be also checked in for the DNA damage repair and apoptosis will be explored. And the inhibitor design will lead to the development of a drug candidate against Leishmania donovani parasite, which leads to control strategy for the Leishmania. So at last, I acknowledge my PhD supervisor, Dr. Wahab Ali, and my director, Dr. Krishna Pandit, and other scientists for their help and support, and Department of Science and Technology for their financial assistance, and at last, University of Calcutta for the PhD program. Thank you so much.
Great. Thank you so much for the talk, Parool, and joining us today. And now I will allow any questions.
Yeah, that was a great presentation, and delighted that you're joining us from India. Thank you.
Thank you so much, ma'am. So,
you've implicated, Cfd1 and Nbp35 in this resistance. Is there a quick way to screen before one, you actually treat anybody? And then you talked about targeting. So 2 questions really. One, is there a quick technique that could be used that's relatively cheap? Because lots of folks are on these antibiotics. And secondly, what would you do to actually target those 2?
Ma'am, what was the first question? Sorry.
The first question was, is there, a mechanism whereby before administering that antibiotic, because to look at antibiotic resistance, it takes several days in culture. Is there a technology, a fast technology you could envisage where when this antibiotic is going to be administered, you'd actually look to see if there's resistance beyond culturing by targeting those 2 genes or proteins? By screening.
No, we have only performed the drug sensitivity assay for the culture, and it is a very easy method for to check the tolerance against antibodies.
How long does it take though for the resistance to is it a 24-hour, 48-hour, or 72-hour?
Yeah, it is done for 24 hours, ma'am.
Okay, so that's fairly fast, but with some of these you want to be delivering the antibiotic really quickly. So I guess what I'm suggesting is there might be a quick screening tool to actually measure the levels of Cfd1 and Nbp35, but that's speculation. And my other question was, how would you actually target? You talked about a therapeutic strategy. What are you thinking of?
Ma'am, actually we want to break the interaction of these 2 proteins, because interaction is very important for these 2 proteins for the biogenesis of iron sulfur cluster. That's why we are planning to design the inhibitors against these 2 proteins. So inhibitors will bind to any of these proteins and interaction will be inhibited. After that, we will check the if the when inhibitor will design, then we will check the inhibitor on the with the treatment of parasite.
Awesome, thank you very much.
And for their survival and growth will also check.
Thank you.